Rice Plant Disease Detection and Classification Techniques, a Survey. Abstract rice/paddy is the staple crop of India. India has the largest area under rice cultivation that includes the cultivation of brown and white rice. Rice cultivation brings employment and also helps to stabilize the gross domestic product, GDP, by its vast contribution. In the field of agriculture and modern computer era, recognizing infection or diseases using plants' image is one of the keen research areas. This research paper provides a survey on various techniques and briefly discusses significant aspects of different classifiers and techniques used to detect rice diseases. Papers of the last decade are studied thoroughly, including the work carried on various rice plant diseases, and a survey is presented based on essential aspects. The survey focuses to distinguish different methods based on the classifier used. The survey gives insights of the different techniques used for the identification of rice plant disease. In addition, a hardware prototype and model using Convolutional Neural Network, CNN, is proposed that detects rice disease. It further identifies the type of rice disease into rice blast, rice blight, brown spots, leaf smut, tungros, and sheath blight. Keywords: rice leaf disease, feature extraction, SVM, KNN, CNN, multimedia sensors. Introduction. India is known for its agriculture as different types of crops are cultivated here. Rice is grown in nearly every state of India, with West Bengal having its highest production, followed by Uttar Pradesh and Tamil Nadu. India's more than 3/4 population depends on agriculture. Crop plant sickness or climatic changes can bring starvation. 1. This may have terrible effects on India's economy too. Hence identification of such diseases as early as possible becomes very important. Earlier manual examination of the leaf was the only approach for disease identification. This was carried out by manual inspection of plant leaves or by referring a book to identify the disease too. 3 major disadvantages of this method are: first, low accuracy; second, it is not possible to examine every leaf; and lastly, it is a time-consuming process. With advancement in science and technology, various ways are invented for identifying such diseases with high accuracy. 2 approaches include image processing and deep learning. Image processing exercises different techniques like filtering, clustering, histogram study to detect the damaged region, and image processing algorithms to identify the diseases. On the other hand, in deep learning, neural networks are used to detect the diseases. Rice diseases are classified into 4 major types: bacterial diseases, fungal diseases, viral diseases, and disorders. Bacterial infections include bacterial leaf streak, bacterial blight, grain rot, foot rot, and pecky rice. Some types of fungal diseases are brown spots, black horse riding, leaf blast, false smut, and any more. Rice tango, grassy rice stunt, rice yellow mottling are few types of viral diseases. Cold injury, white tip, alkalinity, and bronze are a few types of disorders in rice plants. 2 major reasons for rice plant disease are: first, fungal/bacterial attack, and second, unexpected climatic change. 3. While dealing with rice diseases, there are few important things we need to consider, like valid data collection, proper monitoring of rice plant, and many more. Collecting samples of the infected rice plant is one of the important and essential steps. This can be done by installing multimedia sensors at different locations of the farm. This helps in monitoring the rice plant periodically. Also, climatic changes and their impact on the rice plant can be recorded and investigated. But this technique also has certain limitations, like regular maintenance of the system, a shadow in the images captured which result in low accuracy. This paper presents different rice diseases detection techniques and comparison is made amongst them on basis of classifiers used. Some common classifier used in recent past are: support vector machine, SVM, k-nearest neighbor, KNN, and artificial neural network, N. All these classifiers are explained in their respective section. Miscellaneous section includes survey whose classifiers are not known. In addition, a novel hardware prototype and convolutional neural network, CNN, model is proposed that detects and identify type of rice disease. The paper organized as follows: Section 2 summaries all the classifiers used for rice plant disease detection with their respective comparison tables. Section 3 contains the proposed work, and the proposed model is elaborated briefly. Section 4 contains conclusion of the paper. 2. Literature review. This section outlines some of the methods used to detect rice disease. Classifiers used in disease detection are considered as the subject of comparison. A. SVM. SVM separates the data sets of the two classes and draws a line, decision boundary, also known as the hyperplane. This hyperplane is drawn considering the extreme points plotted on the graph. Hence SVM is known as best divider of the two classes. 3 common diseases, namely brown spots, rice blast, and bacterial blight, were diagnosed using an automated system. 4. K-mean clustering was used to separate infected part of the image. Pesticides are also suggested for the identified diseases. Also, a computer-based automated structure was developed for disease diagnosing in Article 5. A 21D feature vector is formed by selecting features from infected and non-infected leaf with the help of gray level CO occurrence matrix, GLCM, accompanied by color moments of the leaf. An extended version of the previous method where K-mean clustering method is used to extract details like color, SD, area, perimeter, etc. Components like shape, texture, and color are selected as features to classify these diseases. 6. Another method to detect the rice diseases includes image acquisition that consists of leaves of rice plant of both types, infected as well as non-infected. Images were taken from a village named Sheath in Gandhinagar, Gujarat, India. 7. Image pre-processing consists of obtained HSV model for the RGB color model. As a part of image segmentation, 3 clusters are formed from green cluster, background on the image, and infected part of the image. Further diseases were classified using SVM. Another research work used OS2S method to segment the rice plant leaves. 8. After segmentation, various features were selected with the help of local binary patterns, LBP, and a histogram was drawn. Further, SVM was used as classifier, and this research work successfully classified 3 major rice diseases, which are bacterial leaf blight, leaf smut, and brown spots. An automated system was introduced which employed SVM classifier, which can identify leaf brown spot as per morphological changes of leaf. 9. Here image segmentation incorporated noise removal and computing radial hue of the infected region. Another paper in which performance of CNN model with SVM was employed for rice plant diseases detection using 5,932 on-field images. 10. Also, small CNN models like ShuffleNet and MobileNet2 were tested on the basis of sensitivity, false positive rate, accuracy, and specificity. It was found the ResNet 50 plus SVM provides the best results. Another research employed a method where the diagnosis of the rice diseases was divided into 2 stages in order to categorize leaves into infected or non-infected. 11. In first stage, pre-processing is done wherein features were extracted from the color moment, and second stage focused on dividing the leaves into 3 major category, namely, leaf blight, brown spots, and leaf blast. This was done with the help of features extracted from gray level CO occurrence matrix. The proposed work also tested the accuracy for different classifiers like multi-layer perceptron, Bayes classifier, decision tress, SVM, and K-nearest neighbor, KNN. All SVM methods discussed in this subsection are compared and summarized in Table 1. BKNN. K-nearest neighbor, KNN, is the most straightforward machine learning algorithm. Based on similarities obtained between the new data and existing data, the algorithm classifies the new data into its suitable category. Global threshold method was employed with KNN classifier for diseases identification. 12. Features extraction included calculating length of major and minor axis length, perimeter, and area. Proposed system provided an accuracy of 76.59%. Classification of diseases using minimum distance classifier and KNN was proposed in 13. Information related to shape and color of the infected images were extracted and were inserted into the system to classify 4 major diseases, namely rice brown spots, rice blast, sheath rot, and bacterial blight. System provided an accuracy of 89.29%. Another method wherein 5 attributes are selected using correlation-based feature selection technique of Verekar 14. The process includes extraction of crucial features like color, the area from the leaf, and feeding it to the KNN algorithm. KNN classifier along with decision tree and naive Bayes were used for generating more accurate results. A system that uploads the acquired and segmented image to the cloud using compressed sensing was used in 15. This system is useful to minimize the complexity of manual monitoring in the big farms and easily identify the diseases at a very early stage. K-mean clustering segmentation methods is used to perform segmentation for infected area. The segmented image is reconstructed from the cloud using compressed sensing recovery algorithm. Feature extraction is carried out using GLCM method and is provided to KNN classifier, which successfully detects bacterial blight, sheath rot, brown spots, and rice blast respectively. All KNN methods discussed in this subsection are compared and summarized in Table 2. CN. The idea of N is created by visualizing the human brain. There are 3 layers in a neural network, namely, input layer, output layer and hidden layer. Neurons makes up the hidden layer. Layers are connected via channels. An activation function is generated which triggers a particular neuron from the hidden layer. Number of hidden layers vary with the complexity of the pattern. An automated system that uses machine learning algorithm for classifying leaf to be healthy or infected and further classifying the diseases was proposed in another work 16. Leaves were captured from a distance of 25 centimeters and features like standard deviation, energy, correlation, and many more were used to train the system. 90% accuracy was achieved in identifying the infected leaf. Another research work implemented a light-controlled module to capture images 17. Height and width of the box were calculated by keeping in mind the distance of the object from the camera and the amount of light entering the box. Thresholding and masking were the main procedure carried out on the converted the leaf into binary level image. Diseases like rice blast, leaf blast, and brown spots were identified. Deep CNN is used in identifying diseases in 18. First pre-processing is carried out on the data set of 500 images. With the help of this pre-processed image the CNN model is trained. Experiments results show us that CNN is best classifier in terms of speed and accuracy. Another research work used fractal descriptor for identifying 4 major rice plant diseases, namely, brown spots, leaf blight, leaf blast and tungro 19. Manually extracted images were used for the study. Saturation component was used in classification processes with PNN classifier. Accuracy of 83% was achieved. A mobile application which used fuzzy entropy and probabilistic neural network was developed by researchers in Article 20. Feature extraction is done using fuzzy entropy and are given to PNN classifier, which helps to detect tungros, brown spots, leaf blight, and leaf blast rice diseases. All N methods discussed in this subsection are compared and summarized in Table 3. D. Miscellaneous. An expert team was appointed having 15 normal people and 20 agricultural expert in order to identify the type of diseases 21. This expert system was able to identify few diseases, namely, false smut, rice bug and sheath rot. Identification process was carried out via features obtained from the leaves like color of spot, shape, size, infected part etc. Another method where various segmentation methods were carried out with normal and abnormal leaves. Implementation work was carried out using MATLAB 7.5 Tool 22. It was found that region growing algorithm works best as a segmentation technique. An experiment on artificial inoculation rice blast was conducted in Article. This was carried out on 3 locally grown varieties of Japonica rice 23. A push broom system was designed in order to capture the leaf images. Conversion of reflective values to DN values was done and the result was given to Prokut system for classification of the diseases. Another author used Euclidean distance as his classifier to identify the rice diseases 24. First, segmentation of images was done using k-mean clustering algorithm. Then GLCM algorithm was used to extract texture features and metric and eccentricity algorithm was to trace the shape pattern. Obtained information was given to Euclidean classifier for further analysis. With the help of PHP and MySQL, an expert system was designed to identify the rice disease 25. This study was carried out in Meghalaya, India and had three major sections. First, to identification of common rice diseases in the state. Second, to represent it via computer program and lastly demonstrating the expert system to the common people. Another study was carried out wherein the images of the rice plant were captured via smartphone, further transferring it to computer for RLB detection 26. The RLB disease severity stage was displayed as output and this information was used by farmers and agricultural expert for detail study. Another disease detection techniques include the classification of diseases based on the RGB percent using image processing 27. This technique takes the RGB percentage of the infected leaf into consideration for disease detection. Based on the RGB percent value, leaves are grouped into different classes. Further, this information is fed into Gaussian naive Bayes, that categories disease into different categories. Advantage of this methods is that it requires less time to identify the disease, but identifying diseases based on RGB percent may produce a less accurate result. An optimistic fuzzy interface system was designed for automatically identifying the rice disease 28. Pre-processing includes removal of converting the image into RGB band and using median filtering to remove noise from the green band. Then the color features and textural features are extracted from the green band which are given to the OFIS system which classifies the leaf image as infected or not infected. All other methods discussed in this subsection are compared and summarized in Table 4. 3. Proposed method. As shown in Fig 1, the proposed method for the identification of rice disease has the following steps. Data acquisition. The collection of the rice plant images with other soil and environmental parameters is the first step. In proposed work, hardware module comprises of Raspberry Pi board with scalar and multimedia sensors. Solar panel is used for energy harvesting. Multimedia sensor, camera, is mounted on metallic pole and the vertical movement is controlled with the help of motor. Scalar sensors like temperature, soil, humidity, moisture are used to collect scalar data which can be used along with image data for further processing and analyzing. Scalar and image data is collected periodically and uploaded on the cloud to monitor life cycle of rice plant. Pre-processing, collected images may be irregular in nature, so pre-processing is carried. Here RGB colored images are preferred over HSV because of their clarity and also conversion of RGB to HSV may be time consuming. Further, feature extraction process is carried out, which are then given to the classifier. Classification, the CNN classifier is used to classify whether the leaf is infected or not and then identify the diseases accordingly. In proposed work, Python-based computer vision and machine learning libraries such as Pandas, NumPy, TensorFlow are used for processing the received data and identification of the diseases. With the help of CNN model, an outstanding accuracy of 96% was achieved for classification of major diseases namely rice blast, rice blight, brown spots, leaf smut, tungros, and sheath blight as shown in Fig 2. For conclusion. Rice disease is the major issue encountered by most farmers, hence its early detection is very essential. With advancement in science, identification of rice disease is much easier than manual inspection which was carried out in earlier days. This research paper summarized various methods used for identification of rice diseases based on the classifier used. Also, it was found that CNN classifier holds outstanding record in pattern recognizing problem which is core concept of image processing. Our proposed model that works on CNN shows promising results in achieving good accuracy. In future work, we want to compare our CNN model with existing state-of-art techniques. In addition, we want to customize our hardware prototype and CNN model for other types of crop plants.